Okay, hello everyone. This is my presentation regarding immunoprecipitation, which is isolation protein technique. And let's get started a little bit. So here's a little bit of an overview about immunoprecipitation, or what is referred to as IP. And so basically what it does is it tries to separate antigens or any other proteins that are within an antigen antibody antibody containing group and try to isolate them in order to analyze them using mass spectrometry or SDS page to get more information on size, particle interactions. And here's a little bit of an overview on the right that I got from the textbook image regarding the processes of immunoprecipitation. Now, it's really simple. It's a cell extract containing stable protein complex, and then that protein complex is bound with an antibody. And then this isn't really detailed, but it does a SDS separation page in order to get more information regarding the protein size, mass of the proteins when it's degraded, and also the identification, whether it be the mRNA component, genetic component of that protein. And this is just a simple overview, but I'm going to go under the in-depth processes. So in order for this immunoprecipitation to occur, an antigen must bind to an antibody, a primary antibody. And in order for that primary antibody and antigen to bound with each other, there needs to be a form of method of separation. And there they use a within this uh, diagram, they use two forms of separation, which is the agarose beads or magnetic beads. And I'll go over in depth the advantages of one over the other. But this agarose bead and magnetic bead, what is interesting about this is when the antigen and the primary antibody are bound in a seed full of antigens or other proteins involved, this allows for a separation from that whole containing group, which can be utilized in cells, tissues, or different systems within our body. And once they're bound, they go through an elution process where now it becomes separated with the antigen and the antibody all together after the elution process, and then is put into an SDS page or Western blotting. And SDS page or Western blotting, what it is essentially is Western blotting, as we know as snowdrop, is a protein analysis, whereas SDS page is a separation analysis as well as a protein analysis that emphasizes the size of the protein spectrum. And there is a difference between agarose and magnetic bead system in that agarose is the simply bound 100 micromolar bound of the antigen and the protein, antigen and antibody protein. Whereas magnetic beads, obviously, as with the name, utilizes a magnetic approach, which it has a much greater advantage compared to agarose. And they also have a magnetic agarose beads. And the advantage of magnetic beads is that it has a greater yield, greater ease of use and speed, and can be able to, within the whole sea of strands of various antigens and proteins that are embedded within our bodies, it is able to isolate each individual or an individual protein that is specific to our needs of analysis. Whereas agarose, there's a little bit of a disadvantage in that it's very slow and it is not as reliable as a magnetic bead. Now, there is a very big importance of using immunoprecipitation as compared to other forms of protein isolation analysis, compared to column chromatography or even SDS page or Western blotting, in the sense that this is one of the most widely used methods to isolate proteins from either the cells or tissues within our body. And as you know, there are various proteins that are embedded within us. And in order to isolate an individual protein or strand, it is very remarkable. And what this does is that this not only utilizes the immunoprecipitation process of an agarose or magnetic bead attaching to uh, antigen or antibody containing cell or group, but what it does is it binds to that and also incorporates SDS page or Western blotting, which are another protein analysis processes, in order to analyze each individual protein that was extracted. And that is really cool because immunoprecipitation can essentially go throughout our body, whether it be our immune systems, particularly our immune systems, to analyze an antigen or any other proteins that play an essential role into different systems of our body, whether it be the nervous system, cardiovascular system, immune system, you name it. And it also plays a role of the biochemical components of our body. Now, a prime example that is used today is actually a discovery that was investigated within the year 2020, and it discovered a protein called 1011 translocation 1 or TET1 protein. And I didn't know about this until now, but it is a protein within the oligodendrocytes of our body, which is oligodendrocytes are important factors for myelin sheath within the central nervous system. That's what oligodendrocytes are used for. And through immunoprecipitation, they were able to analyze the mass of this protein and its ability and role of what it does within oligodendrocytes. And it's involved in the homeostasis and differentiation for the myelin sheaths of the central nervous system. So it plays a very important role in the structural and metabolic component of the oligodendrocytes. And what it does is actually really cool is that it initiates a demethylation pathway in order for differentiation to occur within the oligodendrocytes. And even though that this protein went through numerous trials and numerous studies through Western blotting SDS page, the ability to utilize immunoprecipitation as well as co-immunoprecipitation to further in-depth analyze it with Western blotting and SDS page showed a new discovery because there are various 1011 translocation proteins, but TET1 metabolically as well as structurally played a significant role in the oligodendrocytes homeostasis and differentiation. And yeah, and this was one of the prime examples of this immunoprecipitation being utilized within our biochemical pathways in our body, especially within the central nervous system. And here is my works cited with the textbook as well as various published articles that I found online. And thank you so much once again.